Okay, can somebody confirm you can hear me?
I can hear you.
Great. Actually, let's get rid of that. Come on. All right, today is April 24th, and we're covering Lab 5, Streaking for Isolation and Culture Media. Any question about anything? I thought I had that. There we go. Lab 5, Cultivation of Bacteria, Culture Media, Inoculation, and Isolation. Any questions? There is a little bit of reading to do before you begin this lab. We do have some video clips in this lab, but the virtual lab no longer works, so it's been removed. The virtual lab we used to have worked on Flash, and if you don't know about Flash, it no longer works. The learning objectives for this lab is one, be able to understand the importance of proper specimen collection and transport to the lab. Two, explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between the various types of culture media. 4, explain the purpose of streak plate culture technique, aka streaking for colony isolation. And then 5, define the following terms: selective media, differential media, and reducing media. Any questions about what we're going to be doing? Move that over a little. Whoops, too far. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they'll dry out, and others to exposure to oxygen, like most anaerobes, and thus require a special transport medium to keep the specimen alive. Once a specimen is received in the lab for microbiological culture, microbiologists employ the 5 principal I steps to process it. 1, inoculation. 2, isolation. 3, incubation. 4, inspection. And 5, identification. Inoculation is placing the specimen on an artificial culture media to initiate its growth. 2, isolation, you need to spatially separate out microbial growth into distinct colonies. 3, incubation, you have to place the culture medium in an environment with conditions to facilitate the growth of the culture. 4, there's inspection. Upon examining the culture for the presence of microbial growth, you may need to subculture it to get it isolated. And then 5, identification, that is performing specific tests to ID any pathogenic microbe that may be isolated. These 5 steps will be described in different types using different types of culture media. So the different types of culture media, you are already familiar with general purpose media. That would be NA, TSA, or NA, that's NA with the nutrient yeast extract added to it. These general purpose media are media that do not select for anything. They're just cheap and they grow a lot of microbes, but they do not grow fastidious microbes. To promote the growth of fastidious microbes, microbes that have specific nutritional requirements, you must use and enrich general purpose media, like blood agar, that's 5% sheep blood, added to the media to get the organisms to grow. And blood agar, or BAP, is a enriched general purpose media, and so is chocolate agar, or CAP, enriched general purpose media. Any questions about any of that? And then we have selective media. These media contain chemicals that inhibit or halt the growth of some organism, but not other organisms. Keep in mind two things when using selective media. First, a selective media isn't necessarily perfectly selective, meaning it's not 100% selective. Given enough time, even organisms that are inhibited may reproduce enough to form visible colonies. And second, even those organisms that are supposed to grow on the selective media may form smaller colonies or may grow slowly, more slowly on the selective media than they would on the general purpose media. Any question about any of that? An example of a selective media is 7% sodium chloride media. If the organism cannot grow in 7% sodium chloride, then it will not grow and will be selected against. Any question about any of that? All right, the next media I want to talk to you about is differential media. This media contains chemicals that produce visible differences among different groups of organisms, such that when they're grown, the differences can be distinguished. They may have different colors, they may have different, I don't know, not shapes, but different, some other difference, different aspect, a different aspect around the colony, for example, a different color. These differences are unrelated to how well the organism grows on the differential media. Differential culture media allows you to distinguish between dissimilar bacteria by visually distinguishing the two species from each other. Lastly, we have reducing media. They promote the growth of anaerobic bacteria because low or no oxygen conditions are required to grow anaerobes. So reducing media removes oxygen from the media, and then you can grow anaerobes. It's important to note that a culture media may be classified into more than one category. For example, McConkey's agar is both selective and differential. Blood agar is both an enriched general purpose media and a differential media. Any question about any of that? So how we're looking at McConkey's agar, it is selective in that gram-positive cells tend not to grow and gram-negative cells tend to grow. So if you get growth, it's most likely gram-negative, but it's also differential because if there's growth, you can differentiate between the organisms that can ferment the sugar lactose. They will appear pinkish, and the background will appear pinkish as well, from those that cannot ferment the sugar lactose, and they will be their normal color. The background is its normal color, and then the organism is either white or off-white in color. So that's McConkey's agar. Here's blood agar. It is both differential, allowing you to differentiate between different forms of hemolysis, and it is an enriched general purpose media, allowing you to grow fastidious organisms like Streptococcus Pyogenes will not grow in a general purpose media. It needs to have certain additives added before Streptococcus pyogenes will grow. If you add red blood cells in blood agar, you are adding the ingredients required by Streptococcus pyogenes, meaning the blood supplies it, and Streptococcus pyogenes can grow on blood agar. So it is an enriched general purpose media, and it is also differential. Here we're looking at hemolysis, gamma hemolysis or no hemolysis, where the red blood cells are not being lysed when we look at the blood coming up to the cells. Here we're looking at alpha hemolysis. We got that on the other side. Here we're looking at alpha hemolysis, where the red blood cells are partially hemolyzed, meaning they're partially lysed. And that's why we're seeing a dark ring around the off-white smear of the bacteria in the alpha. And inside the alpha, it's also dark. And that's from the partial hemolysis. Partial hemolysis is usually darkening of the blood or a greenish color of the blood around the cells that cause the alpha hemolysis. Now here we have the bacteria shown in the beta, and this bacteria is white. And then the region around the bacteria is clear. You can actually view through that region right there. So you can hold this plate up to a light and see the light bulb underneath this region, but you can't see through the blood. You can't see through the bacteria, but you can through the beta hemolysis, where the red blood cells are completely lysed. It is normal to see alpha hemolysis surrounding beta hemolysis. Any question about any of that? Oh, good. The lecture file finished. So my computer will now start working a little faster. Go ahead and name that now. Oh, come on. All right, this table is showing you the different types of media, general purpose media, including tryptic soy agar or TSA, nutrient agar or NA, or nutrient agar with yeast extract or NA. And then a differential and enriched medium, that would be BAP or blood agar. There's also just an enriched general purpose media, that would be CAP or chocolate agar. And then there's selective and differential media, that would be MAC, EMB, and HE. All three of these select for gram negatives. They select against gram positives. All three of them are differential, allowing you to distinguish between cells that can ferment the sugar lactose and those that cannot ferment the sugar lactose. So if the bacteria can grow on the McConkey's plate, it'll have a pink color from its ability to ferment the sugar lactose. EMB, the ones that can ferment the sugar lactose, will look either greenish or metallic. What's the word? Metallic green or black. And then HE, if it can ferment the sugar lactose, it will be salmon colored. That's a yellow-orange color. And then there's a selective media. PEA is a selective media. It allows you to select for gram, let's see, for gram positive cells The gram-negative cells are inhibited. And then there's mannitol salt agar, which is both selective and differential. It is selective for cells which can tolerate growing in salt, 7% sodium chloride in MSA, and it is also differential in the ability of the cell to ferment the sugar mannitol. If the bacteria can ferment the sugar mannitol, it'll be yellow and there will be a yellow halo around the colony. If it cannot ferment the sugar mannitol and the bacteria grow on the plate, meaning it does tolerate the 7% sodium chloride, then it will be the bacteria's normal color, whatever the colony normally the color looks like, usually white or off-white, but it could depend on what species we're using, what color it has. Anyways, those are the different media in the table. You can use this table for the lab. The 5 I steps. 1, inoculation. That's putting the bacteria you obtained from the clinical specimen on a plate to grow the specimen. And the plate you use will depend on what you're looking for. We'll talk a little bit more about that later. And then isolation, that's isolating out the species of interest. Louis Pasteur developed the concept of growing a pure culture, that is a culture containing one and only one species. And he had a step for isolating a pure culture. It's a very difficult step where he grew his cultures in liquid culture and he made different serial dilutions of the culture. And then he would replica plate each serial dilution. And if ever he got a dilution growing where only one tube had growth in it and 9 tubes were no growth in it, he correctly concluded that the one out of the 9 tubes that had growth in it was likely a pure culture. Robert Koch developed a much easier way of obtaining a pure culture, and that is he streaks out his mixed culture on a solid media, takes an isolated colony from the first plate, and then streaks it again a second time to get isolated colonies on a second plate. And then he takes the cells that have gone through 2 colony isolation events and grows those cells up. And then he is most likely going to have a pure culture. It's not 100% guarantee, but over 95% of the colonies that go through 2 colony isolation events from plate streaking will be a pure culture. And over 95% of the cases is good enough for microbiology. There is no easy way to get 100% pure culture. So this is what we use. And actually, Koch's method that he developed is so good, we still use it today to get our pure cultures. So theoretically, when you're streaking out a culture, someplace, either in the first quadrant, the second quadrant, the third quadrant, or the fourth quadrant, someplace you'll be putting down isolated cells, which will then grow up into isolated colonies. And if you do that 2 times, get your cells that go through 2 colony isolation events, you will most likely have a pure culture. And here we're seeing streaking on a plate. This is not quite correct, but it's close enough. You take your original sample, streak it out on quadrant 1, and you should streak it out actually 20 to 30 times. You can overlap the place when you're streaking 20 to 30 times, which as you can see, they're overlapping here. It's not quite isolated like that. And when you streak out, it'll be very heavy on the first streak or 2, And then it'll be a little bit lighter because you're getting some dilution as you're streaking out the plate, so that this region will be lighter than there. And then you sterilize your loop to remove the bacteria from the loop. And then you start streaking out too. And this is not showing it correctly. You actually put it in quadrant 2 and then move it into quadrant 1 where the bacteria are lighter on the streak. So you'd bring it here, maybe as far as here, but you don't want to go all the way. Actually, you wouldn't want to go here because that's only the second streak there. So you'd want to go here and you bring it back about 3 or 4 times to pick up cells from quadrant 1 and carry them to quadrant 2. And then you streak that out about 3 or 4 times. And then you sterilize your loop, put it on quadrant 3, which this is actually shown correctly, and then you bring it into quadrant 2 to pick up the cells, but you don't go to here, because this is where the bacteria of quadrant 2 are their heaviest. You only go where the bacteria are light, on that streak, maybe that streak. So you go like to here, and then go back into quadrant 2 3 or 4 times. And this is going in there too many times. It's about 6 times. You don't do it 6 times, you do it 3 or 4. And like I said, you don't go all the way to here. You'd only go to about here to pick up your cells from quadrant 2 and then streak it out on quadrant 3. Quadrant 4, they've actually done correctly, and that is you then sterilize the loop, put it on quadrant 4 to cool it, bring it back to quadrant 3, bring it back about 3 or 4 times, and you do it where the quadrant 3 is lighter, not where quadrant 3 is heavy. And then streak it into quadrant 3 3 or 4 times, streak out quadrant 4, and then someplace along the line, you will be plating individual cells which will grow into individual colonies. In this case, it happened in quadrant 4, but if it was more dilute, it would happen in quadrant 3. More dilute, it would be in quadrant 2. And if the stock culture is very dilute, you could streak out individual isolated colonies on quadrant 1. The point is you just want isolated colonies. And then you take an isolated colony and you streak it out on a second plate to get isolated colonies. And then on the second plate, you pick out an isolated colony and you take those cells and grow it up. And that would be most likely greater than 95% chance a pure culture. Any question about any of that? All right. So incubation is putting the culture in an environment that will promote the growth of the bacteria to grow. Usually we put our plates and our cultures in an incubator. In our microbiology lab, we only have simple incubators that simply heat. We add humidity. That bucket there has water in it that adds humidity. The humidity is important because the plate can stay in an incubator for 1 to 2 weeks before drying out, meaning the cells will stay alive for 1 to 2 weeks in a incubator that has water for humidity. If you don't have the humidity, the plates will dry out after about 3 or 4 days. And then the cells will no longer be alive, assuming they're normal cells like E. coli. So our simple incubators simply control the temperature. You can set it at different times. We have one at 37, maybe some at 35 and some at 30 degrees. And they control the humidity, meaning you just put in some water and that puts the humidity in the incubator. And then you allow it to grow for a certain length of time, which for our microbiology lab is usually 2 labs, but 2 days, but it could be 1 day. It could be an entire week if they're very slow growing. and then you pull out the plate and take a look at it. This is a much more complex incubator, and it can control the amounts of oxygen and the amounts of carbon dioxide available to the cells. If we were growing an anaerobe, for example, we want to remove the oxygen and then usually put up the carbon dioxide. So you'd put either nitrogen or carbon dioxide in the incubator. And with an anaerobe, because they'll be sensitive to oxygen, when you open up the incubator, oxygen would get in. You'd probably want to grow the anaerobes in an anaerobic jar, which seals. And then when you open the incubator, the plates will still be anaerobic. If you don't know, the plates will allow air to come in, even if the plate is capped, not capped, but the lid is on, air can still get in a plate. And so just opening up the incubator will allow oxygen to get in. And then it'll go right into anything that's not sealed shut. In the plates, it's almost impossible, not entirely impossible, to seal them like this is obviously sealed. All right, once you've grown up your cultures, you then inspect them, looking for obvious visible colony morphological variations to help you distinguish between the bacteria. Microbiologists can describe the appearance of colonies with a variety of descriptors like size, shape, margin, that's the edge of the colony, the elevation of the colony, to help begin to identify the colony, meaning the species. However, it will not be enough to identify the species. You then have to identify the species by usually performing specific biochemical tests. And that's the way most labs do it. Another way to do it would be to actually sequence the DNA of a pure culture to determine what species it is. Very few microbiology labs do that. Most of them just run biochemical tests. And we'll talk about some biochemical tests you can run in today's lab. Any question about any of that? All right, know the terms of this lab. You can skip the references. In this lab, everything we will be doing will be online, but we do expect you to understand the procedures and the techniques that we're talking about. We have some videos for you to watch. Here's a video on inoculating an agar plate using the streak plate culture technique. You only need to watch it to 4 minutes and 15 seconds into that video. You should note that only 3 sectors are streaked out in this video. It doesn't matter if you streak out 3 sectors or 4. We showed you above 4 sectors. And then here's a video on showing you beta hemolysis, another one showing you alpha hemolysis, and one showing you gamma hemolysis. So watch those videos. And then there's a video talking about McConkey's agar. You only need to watch that video into 3 minutes and 50 seconds into the video. And then a video talking about mannitol salt agar. You should watch that video. Any question about any of that? Let me go to the worksheet now. All right, I think everything we do is done online. So I've got some pictures and tables for you in the lab module. So you'll need to refer to that. But from the questions, you probably only have questions in the worksheet. I answer the question, describe a natural circumstance where Staphylococcus species of bacteria might benefit from tolerating salt. So give me a natural circumstance where the bacteria grow and are exposed to salt, not something like growing it on a 7% sodium chloride agar plate, because that's not a natural circumstance. And then 2, how might hemolysis benefit a hemolytic organism? if we have a hemolytic bacteria, how may hemolysis of blood cells benefit the bacteria? 3. Imagine that you have a culture containing a single bacteria species. This culture may be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to help identify the unknown bacterial species. That is, name 2 properties from each of these plates to help you identify the species. You only get full credit if you name 2 properties to each plate. If you name less, you won't get full credit. You may name more than 2 if you wish, but you only need to name 2. 4. What is the main goal of streaking for colony isolation when you're streaking out a plate? 5. What happens to the growth on the plate if you do everything correctly, but you forget to sterilize the loop in between sectors when streaking for colony isolation? Be specific in your answer. Discuss the growth you see on each sector. And note, this is saying that you're streaking out the plate and you forget to sterilize the loop in between sectors. It doesn't say you forgot to sterilize the loop before you streak out sector 1. So everything is done correctly. That means you are sterilizing the loop before you pick up the cells to streak out sector 1. Question 6. What happens to the growth on the plate if you do everything correctly, but you transfer from stock culture to inoculate each of the sectors on the plate? Once again, be specific, discussing the growth on each sector of the plate. And let me warn you that students often get questions 5 and 6 wrong the first time, but the majority of students get question 5 and 6 correct on the second time. And that reminds me, I need to look and see how you guys voted for grading the labs. 7. Suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species on the plate, meaning you streaked out from a mixed culture, and then you notice the plate has 2 bacterial species growing on the plate. How can you use this plate to create a new pure culture of only one of the bacteria? Be specific in your answer, telling me the steps you do to get out a pure culture. 8. In the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. To note whether each medium is non-selective, a general purpose media, selective, and or a differential media by placing an X in the appropriate column or columns. You may use the table of media in the lab module to answer these questions. So for nutrient agar, if you think it's non-selective, put an X there. If you think it's selective, put an X there. If you think it's differential, put an X there. If you think it's all 3, you can put all 3 X's. Although I'll give you a hint, there's no media that is both non-selective and selective, because these terms are, what's the word? Separate. And do that for tryptic soy agar or TSA, do that for chocolate agar or CAP, do that for blood agar or BAP, do that for McConkey's agar or MAC, do that for EMB or using methylene blue agar, do that for hectone agar HE, do that for mannitol salt agar or MSA, and do that for phenyl ethyl alcohol or PEA. And then 9, which agar plate or plates were inoculated without using streak plate technique, meaning streaking for colony isolation? Plate A, plate B, or plate C? I will be here until 8 o'clock or until the last student logs out to help you with this lab. Are there any questions? All right, get started on the lab.